Okay, so what you're going to do, and I'll already have the template built for you, same as I usually do, okay, is you're going to design a lab experiment that will study the effect of a variable of your choice on the growth of plants. Okay, so you can think tonight about what you want that to be. It can really be just about anything. Okay, I've seen stuff like, what color of light do plants grow best with? I even had one person who talked about the tone of voice people use when they talk to their plant. Does that affect plant growth? Okay, because we all know that talking to a plant makes it grow better. Okay? It's scientifically proven that talking to your plant makes it grow better. You don't actually have to talk to it. You could look, I don't know if you'd look more or less weird, but you can just go up and breathe on it. Okay? It just likes the carbon dioxide, that's all. All right? That's why talking to your plant's good for it. But I've had I've had some very creative ones. Okay, like the tone of voice one I thought was really good because it was just funny to imagine somebody shouting angrily at their plant. Okay, and ironically that would probably make it grow better because you'd be giving it more carbon dioxide from shouting at it. But yeah, so I've had lots of really creative things are variables here to test. But here's the thing that people screw up most often on this. They end up saying they want to test this thing, and then they don't design a controlled experiment and end up testing 2 or 3 things. If that's the case, will you ever know if the thing you thought you were testing causes the change? Okay, you won't. If you don't control everything else that could affect the growth of a plant, and that's why we're doing this. We just talked about the factors affecting plant growth today. Okay, so you have to make sure you design a controlled experiment. Controlled experiment only tests one thing. Okay, you can test variable amounts of that thing. If it's like, for example, the color of light. Okay, there was only one manipulated variable, the color of light, but the person tested like 8 different colors. Okay, that's not 8 different variables. That's one variable with 8 variations. Okay, one person said, they were going to add things to the soil. Okay, so they were going to give all of them the same soil, and one they were going to water with Gatorade, one with Coke, one with regular distilled water, and one they weren't going to water at all. Okay, and these were all controlled experiments. Okay, all they were doing was modifying the soil. Okay, with that experiment, even though they modified it with different things, everything else was kept the same. They all got the same amount of light. Okay, they all had the same soil. It was the same kind of plant, same size pot. All of these things are things that you have to think about when you're designing your experiment because they're what I'm going to look for. Okay, so when you're listing your manipulated variable, you got one of those. Okay, your responding variable can very generally be the growth of the plant, but when you say growth of the plant, you've got to tell me how you're going to measure that. Okay, what is it you're going to look at? If it's if you're testing this on carrots, you might want to look at how big the carrots end up at the end. Or if you're testing this on beans, maybe how much fruit or how many blossoms it has. Okay, if you're testing something simple like bamboo, you could just measure how tall it gets. You have to pick the plant, okay, and you have to decide how are you going to determine its growth. Okay? What thing or things are you measuring? You might use more than one. Okay? Maybe you're going to count the number of leaves and measure how tall it gets. All of these are measures of plant growth, but you need to indicate when you tell me plant growth is the responding variable, it's going to be measured in this way. All right. And then for your controlled variables, you're going to have probably more than 3 for this one. Okay, because we need to cover the other things that could affect the growth of a plant, and you need to tell me how you're going to control them. Right, so I want to make sure they all get the same amount of light, so I'm putting them all in this special greenhouse that I built, and they're all going to get 12 hours of sun a day, or 12 hours of artificial light per day. Okay, whichever. All right, and then if water is going to be a controlled variable, how much water are they getting? Okay, and I understand most of you probably are like, I don't even know how much water a plant needs. It doesn't need a lot. Like if you say 1 liter a day, you're going to drown it. Okay, we're talking like maybe 100 mils a day, depending on the size of the plant. Like those big plants over there probably get about 100 mils a day. I know I only water them like once a month, but they get about 1 liter then. Okay, so yeah, it's not a lot. Okay, they don't need a lot. Megan, you have a question?
We're not actually going to do these.
No, you're not actually going to do the lab. You're just designing it. So the nice thing about this is you're not going to have an analysis. Okay, there's no analysis, there's no conclusion, okay, because you're not actually running the lab. So what you're doing is deciding what the problem is going to be. What is the effect of blah, blah, blah on plant growth? Okay, you'll have your hypothesis, okay, your if, and then statement. You'll have your design, that is, here's the manipulated variable, here's the responding variable, here are the controlled variables, and then you'll have your procedure. You will have to write the procedure. It is in the procedure that people screw up the controlled part of the experiment. Okay, they tell me they're testing this, and then in the procedure, they fail to indicate how they will control all of the controlled variables, or they miss one. Okay, like they say, I'm going to put one plant in the sun, like they're testing, let's say, amount of water, and they put one plant in the sun, and they put one plant in a dark room. Well, were you testing water, or were you testing how much light the plant got? Because you changed both of them. Right? So you need to make sure you control everything except the variable that you are testing, and you need to tell me how you're doing that in the procedure. Okay, so you'd say, here's the setup. That we're going to put the plants all in this box. Okay, the box has, if you're testing amount of light, it would be okay. This box has, 12 individual boxes, each with their own light, and I can control when the lights go on and off. So I can make sure this plant gets 24 hours, this plant gets none. Okay, this plant gets 12, this plant gets 6. Okay, whatever. Okay, but that's how I'm controlling it. The temperature in all the boxes is controlled. It's always going to be 20 degrees Celsius. Okay, so that temperature doesn't affect their growth. All of those things have to be accounted for. So the procedure is going to be the most detailed thing you've done in terms of procedures for this, because usually you don't have to write them. Okay, so you have to design the procedure and talk about how you're going to control everything, what you're going to measure, how often are you going to do your measurements. Okay, if you want to say, I'm going to check it daily, or I'm going to check it weekly, or whatever. Okay, and what are you checking daily and weekly? Okay, what are you looking at in terms of observations? Okay, that'll be it. You don't have to do an analysis. Now, something else that's going to be on here that you haven't done before is something called an abstract. It's going to be before everything, before even your problem. Okay, an abstract is a short paragraph, 3 sentences, that tells me what you know about plant growth, why you're studying this particular variable, and what its possible applications are. All right, so it's like 3 sentences. Okay. The reason you do that is all science has to be funded, and an abstract is an experiment's sales pitch. Okay, when somebody is trying to get funding for their research or whatever, okay, they have to pitch it to companies to fund it. Okay, and your abstract is kind of like your sales pitch. Here's what I know, here's why I'm studying this, and here's a possible application of it. Okay, the people that are funding it, they go straight to the possible application. Can we get money off of that? Can we make money on that later? Oh yeah, we can. Okay, good. We're going to fund this thing. Okay, unfortunately, that's how it works. Scientists very rarely get rich off their own discoveries. Okay, they basically have to sell them, okay, to companies in order just to get the research done. Okay, it's sad, but it's true. Okay. All right. So that sort of makes sense what the expectations are. Okay, so like we said here, you'll have your abstract. Okay. And then you'll have basically your regular setup, okay, detailed procedure that includes descriptions of activities, measurements, observations that will be made, okay, and include a description of how you would analyze your results. So, say after 6 months, all the carrots will be dug up and they will all be measured and weighed and compared, and then, we can draw a conclusion on the effect of blah, blah, blah on the growth of carrots. Okay, something like that. Okay, so that's all you'll have. You won't actually have an analysis. You won't have a conclusion because you're not actually running the lab, which means you could get kind of crazy with your lab. I had one person who said they wanted to test the effect of gravity and zero gravity on the growth of plants. They say, I'm going to launch one up to the International Space Station and grow it up there. Well, that's hunky-dory. You're not really actually running this experiment. That'd be interesting. Okay, what they found is when they grow plants on the International Space Station, they grow in all directions equally. Okay, mostly toward the nearest window, but gravity has zero effect on their growth. There's no geotropism on the International Space Station because they can't detect which way is up and down. So light becomes there. They get fractal almost. It's quite crazy. Okay, so that's what we'll be looking for there. Okay, again, make sure you have your variables in there, okay, and what you're going to use to control. All right, so that's what you guys will be working on tomorrow. Okay, do a good job of that. You'll get it done in class. Well, the idea is that you do get it done in class. I don't want it for homework. Okay, I want to see it done. Okay, questions on that?
So no observation?
No, you won't have any observations because we're not actually running the experiment, right? So you'll just have the procedure, what you are going to record. Okay, so I'm going to record every week. I'm going to, record how much foliage there is, and count the number of leaves and take a picture. Okay, there's your observations. That's what I'm doing. You're not going to have any real observations, but that's what you're going to observe. Yeah. Okay.